All right, 3.2 part 2. All right, and last lecture we got here, yay, talking about what chromosomes are, what plasmids are, prokaryote versus eukaryote, my chromosome numbers, and haploid versus diploid. That is where we end with a suspenseful ending after haploid diploid. All right, part 2, we're going to go through autosomes versus heterosomes, right? Body cells versus sex cells. Gonna look at some karyograms and talk about autoradiography. Woohoo! Hold on to your shorts. Here we go. Oh, wait. Let's go back. Clear. There we go. All right. Okay. Skip through all those. All right. Let's start today with homologous chromosomes. You remember we talked about roots that can actually help us a lot. The root for homologous, homo meaning the same, right? Homologous chromosomes are same, similar, pairing up. We get 2, right, copies, or 1 copy from each, right? So we have a total of 2. We get 1 from mom, 1 from dad, right? Maternal and paternal. And when those chromosomes come together, they pair up, right? So this one might be from mom egg, this one might be from dad's sperm, but when they come together in the newly created zygote, right? So it comes together, we get a complete set of chromosomes, 46, and they pair up. Okay, so these are called homologous chromosomes. So homologous chromosomes are identical in structural feature, which means they have the bands and the patterns at the same exact locations, and they have the genes at the same locations. This is what we were talking about a little bit with, where were we? Oh, in pedigrees today, when we were looking at if there's a dominant and recessive. So mom, right, might carry the dominant gene here, and then my dad carries the recessive, and so we see the one that is expressed. So if we, and then we see the one that is carried. We'll look at that a little bit more with pedigrees when we get into that. I'm sorry, and Punnett squares. But when I get the 2 pair up, it's possible, right, to have 2 different copies, alleles of the same gene at the same like position here, but one that is expressed and one that is carried. Okay. And this is what I'm talking about here. So the alleles may be different, right? So paired chromosomes, different organisms could have different information at those, but when they go to divide or when they become visible, we call those homologous chromosomes. Okay. Autosome versus heterosome. Look at the difference between those. So not all chromosome pairs and diploids are homologous. So we actually have 22 pairs of homologous chromosomes. And then our 23rd is what we call our sex chromosomes, right? So the sex determination, male or female, biological male, female, comes from these 2 chromosomes, which we call the X and the Y chromosome, okay? So going back to humans, talking about 23 pairs, so our diploid number, remember which is 2N, right, is 46. 22 of those completely are called homologous autosomes. And then my 23rd pair is called my heterosome or sex chromosome, right? Hetero, remember talking about homo meaning the same, hetero means opposite. Okay, so 23rd can either be XY or XX. Biological female are XX, and then biological male are XY. Again, we talked a little bit about this when we were doing pedigrees because of sex carried or sex linked traits, right? So if something's carried on the X chromosome, males cannot be a carrier. They either have it or they don't, but females can carry sex linked chromosomes because they have those 2 copies, right? Notice the difference in size, right? So X chromosomes are quite large. They look just more like my autosomal, right? The Y chromosome is very small here, does not carry much. Does not carry much at all. Sorry, males. You just get that 1 good chromosome, X. So then going back to, I'm looking at meiosis. So mom's eggs, right, can carry an X or an X, right? So the only chromosome that mother can give is an X. Sperm can carry either an X or a Y, right? So the biological gender is determined by the sperm, not by the egg. Okay. All right. So everything I just said. You can go ahead and put this into your margins. You do not have to rewrite this. So your X chromosomes here, XX. Here's my mom. That's an interesting, like, why is she standing like that? Anyway, and she's on her tiptoes. And then so female, biological male. I'm not going to re-say all this. I just said it all. But make sure you have something like this in your margins to notate. I remember in biology, my 9th grade year, Mr. Damerall, that was my 9th grade bio teacher. He's told us that females are double crossers, right? If you need some way to remember that. And we don't know why men act the way they do. Sorry, these are kind of I don't know, silly, but I remember them a very long time from when I had bio. Okay. All right. So this is a karyogram, also sometimes called the karyotype. It's when we see visible chromosomes. I'm going to skip this real quick, right? So they actually, oh no, they don't have an actual picture in here. Oh, I'll show you in class anyway. But it's when I get the chromosome profiles lined up from 1 through 23, and they arrange by length. So my karyograms, the karyotypes actually display the chromosomes from that cell during mitosis, right, during the condensed DNA, because remember, they are not visible before them. They're usually used to identify sex or chromosomal abnormalities. So looking at whether or not I have like the third 21st chromosome, which is Down syndrome, can tell mom, dad whether it is male or female before the external sex or organs are visible, right, during an ultrasound, and are also looking to identify, like I said, some genetic abnormalities. All right, so here's another picture of these karyograms examples. You don't need to draw these down, but or write these down. But here is, we would know that it is a male because of the X and the Y. Remember that the Y is way shorter, so I can tell that this is a normal, what we call male. And then this is a female. I have 2 X chromosomes here, but with trisomy 21. So we can see that there are 3 21st chromosomes, and so therefore a total of 47. And this is what causes Down syndrome, right? So Down syndrome is also known as trisomy 21, and we notate that differently: 47 chromosomes, X and XY. Okay. All right. Another way condensed chromosomes are visible. DNA is too compact to give a reliable indicator of length. So while the condensed chromosomes are visible, the DNA is too compact, right? So our scientist of this lecture, John Carnes, utilized something called autoradiography, right? So again, we're looking at somehow tagging, right? What do we look at radiography for? Oh, and when we did the DNA, the centrifugal, doing the what's it called? The nitrogen base pairs. Sorry. Okay, so uses radioactive thymine. Remember last time it was radioactive, what was it? Was it uracil? Oh my gosh, now I can't even remember. It was just a nitrogen. Oh, this was nitrogen. Yeah, that's right. So it was N14 and N15. Wow, it was not that long ago. So anyways, he uses radioactive thymidine incorporated into the DNA. And then the chromosomes are treated with the silver bromide, and the radiation converts those ions to insoluble grains. Woo, will we have to outline this? We'll go through it if we can. But anyways, he was able to then measure the length of DNA by autoradiography by looking at these tagged. Okay, we'll look at this in class. We're not going to watch it right now. So here is that method again to develop a more visible form, right? So treating the cells, extracting the DNA, fixing to slide, immersing in silver bromide so that we have that visibility, right? And then used in a process by radiation to activate those silver ions, right? This is the silver, Ag is silver. Those ions are reduced to metal, and then the ions are removed. I will let you know if you need to know how to do this one like you had with the centrifugal and the N14 and N15. But just know that that is the, for now, the procedure autoradiography that is looking at the developed pictures to look at the actual length of the DNA. Okay, so autoradiography, karyotypes are showing pictures of my chromosomes, right? And my autoradiography is able to show the length of the DNA by using some type of tagging method that we've looked at before. Okay, so why would I need to do that? So why would I even want to know those? So in addition to identifying its length, it can actually have some other helpful uses. If radioactive uracil is used instead of thymine, remember that my uracil is only in transcription, right? Used outside the nucleus. I can actually identify regions of active transcription. So looking at the area where the transcription is occurring because I'm using uracil instead of thymine, right? So for the DNA, it's titrated the thymine, right? For RNA, it is looking at uracil, right? And it's looking at low rates of transcription, high rates of, so actually looking at rates of transcription. So how fast the DNA is being copied and then brought out to make protein. So what does that mean? So why does that, right? So we could actually look at different areas and how often they are used to create those proteins, right? So looking at not very often, very often. Okay. And that, my friends, brings us to the end of 3.2. And this one, making sure that you know how to identify visually and numerically autosomes and sex chromosomes. Why do I use karyograms versus why would I use the process of autoradiography? That brings us to the end. Next time, meiosis. Woo! Have a wonderful day, and I will see you soon.